All right, so here are some questions and problems from Chapter 7. Chapter 7 is all about genetic recombination, and um, so that's what we'll be talking about. And all right, so this I thought I would go over the some of the general questions from Chapter 7, and then we'll do the problems after that. But I'd like to not just answer these questions just straight up, but use them as a sort of jumping off point for doing for as an sort of an introduction to the topic. And so I'll expand on them a little more than just what the questions are all about. Um, so, so this first one is about recombination, and I'm just trying to get my pen so it's just right. Well, we'll just leave it like this for now. See how it works. Trying some new programs, so we'll see how it goes. But genetic recombination is a very specific term in genetics, and it talks about how the genes are shuffled during meiosis, basically. Now, we already know a little bit about how genes are shuffled. We know if we start out with a couple of parents, and let's say we start out with a parent like that, and we'll keep track of some of these genes with different colors and see if I can't try to be somewhat consistent. Then uh, we cross these together. Then the next generation we can get a big A from that parent and a big B from the blue parent there, and we can get a little A and a little B. And so these are the parents, and we can see that big A and big B are coming together, and little a and little b are coming together. So that means that given to this F1, we know where those genes came from. And then what recombination means is how do those genes get reshuffled and redistributed during meiosis. And so we can get gametes like this and like this. And those are the parentals, because that's how the genes started out in the parent generation. But we can also get big A going with little b, and big A going with little b, and we can get what do we got? Little a going with big B. And so these guys here are the parentals, and these guys we call these recombinants. So these are recombinants. And these are non-recombinants, or parentals. And we're looking at the gametes. And the gametes are the product of meiosis. So we're really interested in what's going on in this F1. How are the genes being redistributed? Big A started out with big B, little a started out with little b, now we're shuffling them, and now big A is going with little b, and little a is going with big B. Now there's two ways that we can get recombination. We can get it through independent assortment, or we can also get it through recombination, or through crossing over. So independent assortment, and we've talked about that quite a bit, and crossing over. So first let's do independent assortment. So we know, so in this parent here, we'll have two sets of chromosomes if there's independent assortment, and we'll have two sets of chromosomes from this parent. And this parent has big A, and big B, big B, and this parent had little a, little b, little b. And depending on how those chromosomes line up, so we know that we can get those chromosomes to line up like I showed there. Let's see. Alright, so we got to make those guys red. Or they could line up like that. So we could have big A, and then we'll put big B over here, and little a, and little b. So this is what's going on in this guy. And so the blue chromosomes came from this parent, and the red chromosomes came from this parent. So maternal and paternal chromosomes. And so they can line up in either of these orientations. If they line up this way, we get big A and big B stay together, little a and little b stay together. But if they line up this way, we get big A going with big B. No, big A is going with little b. So let me erase that. Get thrown off by my little calendar popping up there. So we're going to get big A, and it's going to go with little b, just because of the way these guys are lined up. And then on the other side, we get little a, and we get big B. So these are recombinants, and these are parentals. And we're generating recombinants by independent assortment. And we know that in independent assortment that each of these things is equally likely. So the number of parents equals the number of recombinants. And that's actually how we define independent assortment, where if the number of parentals equals the number of recombinants, or if there's 50% parentals, 50% recombinants, or if it's 1 to 1 to 1 to 1, that's different ways of saying the same thing, that means that we had independent assortment. That's the definition of independent assortment. How does crossing over work? So crossing over is another way to get recombinants, and I want to make sure we're doing it the same way. So the big letters are blue, and so we'll draw those again, and we've got big A, big B, cross to little a, little b. We get a F1 that looks like this, and then we're going to get our different gametes like this. And again, because we started out like this, so everything's kind of the same, where these are the parentals and these are the recombinants. But in this case, so now we're going to be talking about linked genes and crossing over. So now our recombinants, they are not due to independent assortment, because the genes are linked, they're on different chromosomes, and we're getting crossing over. So what that means is that the parentals are going to be greater than the recombinants, because getting the recombinants is not equally as likely as getting the parentals, and I'll show you why. And the reason is because the genes are on the same chromosome. So if we get this chromosome from one parent, and we get this chromosome from another parent, and the genes are on the same chromosome, we can get what's called crossing over between these two things. So what we're going to do is we're actually going to break this chromosome and break this chromosome, and then switch these pieces of the chromosomes at the end. So if we get crossing over, then we will get this chromosome will stay the same, the chromatid on the outside will stay the same, but this one on the inside here is going to swap material with the other one. So it'll be about the same, it'll be the same spot. So we often draw that as an X, like we say we had a crossover right there, and that leads to this. And so when that happens, this big A moved over there, and this little a moved over here. This one is still the same, like that. But now when we form gametes, we'll get this, we'll get this, but we'll also get this. Again, these are the parentals, these are the recombinants, but the way that we're getting the recombinants is because we're getting this crossover between here, and we're switching positions. But whereas in independent assortment, the way the chromosomes line up is random, and so in any given meiotic division, they can line up in one way or the other. You get your gametes in equal proportion, 1 to 1 to 1 to 1. In this case, most of the time you don't get crossing over. So most of the time you don't get crossing over. So what happens if you don't get crossing over? If you don't get crossing over, you just get parentals. So no crossing over, you just get these guys stay together and these guys stay together. And we'll look at another question that shows that. But sometimes you get crossing over. And when you get those, you get recombinants. And because most of the time you don't get crossing over, you get more parentals than you do recombinants. And this is the definition of linkage. I think that went outside of my... So if P is greater than R, parentals greater than recombinants, genes are linked. No independent assortment. Alright, so that was kind of a long answer to this question, but that's what's going on. So, but just to remind you that even if it is independent assortment, we still can get recombinants. But so in a test cross for two genes, what types of gametes are produced with complete linkage, independent assortment, and incomplete linkage? Alright, so we have a test cross. We all know what a test cross is. Pause there. So we've got a test cross. So what would we get? What kinds of gametes would be produced if we showed complete linkage, independent assortment, or incomplete linkage? So what do those things mean? Well, we know what we get with independent assortment. So independent assortment, we get this. And we get 1 to 1 to 1 to 1. Alright, so that's independent assortment. Question A. Complete linkage means that these genes are on the same chromosome, so we draw them like this. And we they're so close together, so they're so close together this way, that we never get crossing over between them. And so that means we can get this, and we can get this. We get all parentals. Here, parentals equals recombinant, so we get 50% parentals, 50% recombinants. That's the definition of independent assortment. Incomplete linkage means we get crossing over some of the time. And so then we get these kind, and we also get recombinants, but we'll get the recombinants less often than we'll get parentals, or in other words, parentals more often than we get recombinants. So So we get recombinants less than 50% of the time. So that's incomplete linkage. And we can actually use this information. We can use the percent of recombinants to get what we call map distance. So we can start to generate genetic maps of how far apart chromosomes or genes are on chromosomes by looking at how much recombination occurs between them. All right, so that's what's going on with that. So what is the frequency of recombinant gametes? Why is the frequency of recombinant gametes always half the frequency of crossing over? So this one's certainly not obvious. So let's take a look at a couple of chromosomes. And I'm going to start to draw these chromosomes sideways because it's easier to sort of visualize crossing over, or at least I think it's easier. All right, so let's put some genes on here. And we'll try to keep our colors consistent. Easier said than done. So we'll put our big A here, big B here, big A here, big B here, little a, little b, little a, little b. Alright, so if there's no crossing over, we just get this. So, and what would we call these? So these are parentals and these are parentals, so we get all parentals. But if we do get crossing over, we'll make our crossover in green. So we draw a crossover like this. That means that that's the spot where the crossing over is occurring. If we get crossing over, we get these guys going together, and then we get this A, but then it actually links down and picks up this little b down here. So when you get your crossover, you get this and then this, and then you get your little a, and then it breaks and gets rejoined up there and goes with big B. So we'll get little a and big B. And then the chromatid down here, so this chromatid does not undergo crossing over. So crossing over only involves, or usually involves, crossing over involves 2 chromatids at a time. Now if we were to measure how many of these we got, even if we get crossing over to occur 100% of the time, every time crossing over occurs, we get some parentals and some recombinants. In fact, every time crossing over occurs, we get 50% recombinants and 50% parentals. Now that's a little different than the 50-50 we were talking about before. The point here is that in an individual crossing over event, even when we're generating recombinants, we also get some parentals. So even if you have genes that are super far apart, so that crossing over occurs 100% of the time, the most number of recombinants that you can get out of that cross is 50%. And so that's why the frequency of recombinant gametes is always half the frequency of crossing over. So if we had a situation where we got crossing over 20% of the time, that means this would occur 20% of the time, but every time it occurred, we'd only get half of the gametes would be recombinant. So if we had crossing over 20% of the time, the percentage of our recombinants would only be half that, or 10%. So that's what's going on there. And it takes some thinking about the chromosomes, and that's a lot of the reason that we talk about recombination, is it gives us a good excuse to really think about the chromosomes and what's going on during meiosis. And it's one of these things that takes practice. This is just sort of a simple definition thing, um, but it's important because it comes up a lot and it makes us think about uh, chromosomes in a different way. So let's say we've got a dihybrid, and if those genes are linked, they could be linked in a couple of different ways. So this could mean this, but it could also mean this. And so, just to answer the question, this is what we call coupling, and this is repulsion. Repulsion just means essentially that the same kinds of alleles are on opposite sides of the line. So the capital alleles tend to go apart, and the recessive alleles tend to go together. In coupling, the dominant alleles tend to stay together, and the recessive alleles tend to stay together. So we call that coupling. So where does that come from? Well, this is a dihybrid, and in this case, our parents could have been this. Now my little color scheme's not in play here, but if we start with parents that look like this, that means big A and big B are starting out together, and little a and little b are starting out together, and then the if we drew it out showing the linkage relationships, it would look like this, just that big A and big B started together, little a, little b started together. But we can also generate a dihybrid by doing this, using different parents, and then that would give us this. So big A, little b starting together, little a, big B starting together, and they tend to stay together. Then when we generate gametes, we can get these kinds of gametes. We can also get recombinants, but in this case these are the parentals and these are the recombinants. In this case, these guys are the parentals. And these guys are the recombinants. Once again, this is the kind of thing that confuses students all the time, because you just get used to thinking that anything that looks different like this, like these guys, they have to be the recombinants. These guys over here. But it's not the case. It depends how you start out at this point, this F1. Alright, so that's what coupling and repulsion configuration means. And the way it affects whether they're coupling or repulsion, how does that affect the genetic cross? It affects the number of gametes you get. And if the genes are linked, then you'll always, in this case, you'll get more of these. In this case, you'll get, I'm sorry, one of these has to be parentals, so these guys are parentals. And these guys are recombinants. You'll notice I wrote these out a little bit different. I tend to write the parentals first, because if I know this. But sometimes you don't know ahead of time which are which. So you just look at the outcome of a cross, and whichever one are more, those are the parentals. And that's always the case. And so we'll get to that in a minute, how you would see this in a test cross. But all you need to know is look at the data of a test cross. Whichever two categories are the most, those are the parentals. Whichever the two categories are less, those are recombinants. And you can get your different kinds of parentals and your different kinds of recombinants depending on where you're at this stage. So how would you test to see if two genes are linked? This is always going to be the same thing. We've got something that's heterozygous for two genes. We do a test cross. Then we look at our different kinds of gametes. And some of those have to be parentals and some of those have to be recombinants. We won't really know until we see some numbers, but if the parentals and the recombinants are equal, then they're not linked. So then we might not even know which ones are parentals and are recombinants, but if these basically give us 1/4 to 1/4 to 1/4 to 1/4, then they're not linked. Or another way to say it is, if recombinants equal 50%, they're not linked. If the percent recombination or percent recombinance is less than 50%, genes are linked. Or in other words, if the parentals are greater than the recombinants, the genes are linked. Or if the recombinants are less than the parentals, and we mean significantly different. And to test that, we would do a chi-square test to make sure if we're going to test whether or not something's linked, we can look and see if they fit a 1-to-1 to 1-to-1 ratio, and if they don't fit that, then they are probably linked. What's the difference between a genetic map and a physical map? Well, everything we've talked about so far have been genetic maps. A genetic map is recombination frequency. It's also called a recombination map sometimes. It's just based on recombination frequency. So if you've got gene A and B, and we do an experiment, and the recombination percent equals 10, then these are 10 map units apart. If we map A and C, and we get 5, then those are 5 map units apart. If we map B and C, and we get 15, then they're 15 map units apart. And then we could develop a map where A and B are 10 map units apart, A and C are 5 units apart, but C is farther away from B, so we got to put C down here. So we'd have A and B is 10, A and C is 5, and B and C is 15. Don't worry if you don't get exactly how I came up with those maps right now. We'll do some problems that are like that, but I'm just showing you that's what we mean by a map. We just mean the relative positions of these guys relative to each other, and that's all based on recombination frequency. So it's sort of more about how you actually figured it out than anything else. So that's what a recombination map is. But they figured out all that stuff before they even knew that DNA existed. And so after we had these maps, and people started working with DNA, they started coming up with ways to figure out where these genes were on the actual DNA. And so this requires something called genetic markers, or molecular markers actually. So you have something on some actual, so this is the actual DNA. And so that's basically the difference, that we can actually map where these genes are relative to specific molecular markers on the DNA. And so let's say that these are restriction enzyme sites. You guys have done some restriction enzyme digests in your lives somewhere along the way, and we can map where these restriction enzyme sites are. So these are just DNA sequences that we can tell where they are. And then if there's a way that we can say, well, gene A fits in between these two sites, and gene B fits in between these two sites, that's what we call a physical map. So it's a physical map relating the position of genes to the physical position on the DNA. And so this is a genetic map. This is a physical map. And there's kind of one other kind of map we can talk about, which is kind of in between these two, is if you have a chromosome, you can actually see banding patterns sometimes on chromosomes, and we'll look at some of these maps later. And so sometimes you can put genes on the chromosome based on what the chromosomes look like under the microscope, and we call that We call this a cytological map, or maybe cytogenetic. So when you see cytology, cytological map, cytogenetic, we're talking about a chromosome map. So genetic is really sort of an abstract map, just based on the results of a test cross, or based on recombination frequency, however you happen to figure that out. And then a physical map means you had to do something with the DNA. You had to use some kind of molecular biology technique and sort of map it. Eventually, you would get the DNA sequence, and you can say, if you know the DNA sequence, and you can map a gene to a particular sequence, that's another example. That's sort of like the best physical map you can have. So physical map relating to DNA, cytological map mapping to a chromosome, genetic map mapping to a sort of just a recombination frequency distance. And because those are different methods, they don't always line up, they don't always match up precisely. So why do calculated recombination frequencies between pairs of loci that are located far apart underestimate the true genetic locus, or true genetic distance between loci? This is related to that other question where they were looking at chromosomes and looking at what happens when you get recombination between them, and why do we get 50% of what we expect. And so if we have these genes, we got some genes up here, and so these are chromosomes. Each chromosome has 2 chromatids, and on the chromatids are some genes, and we're going to get some crossing over. So we'll make our crossing over green again. So if we get crossing over, then we would get parentals, and so we'd get some of these, we'd get some of these, we'd get some of these, and we'd get some of these. So these are parentals, these are recombinants. And the way you visualize this is you say, okay, this chromosome, you kind of follow the chromosome along, and then when you get to this X, that's a crossover. And so that means that we're going to break this chromosome, and we're going to break this chromosome, and then these 2 bits here are going to switch places. And so what that'll end up with is this big A will now be connected to this little b. So that's how you draw these chromosomes. Alright, so that's cool. And so if you get a crossover, then you start to generate some of these recombinant gametes. You count them up and you can figure out the genetic distance between those genes. But as the genes get really far apart, let me make sure I got the right color. And I'll draw them the same distance, just because that's all the room I have. I'll put the genes back on there. Put some blue genes up here. Try to avoid any kind of blue gene joke. And now, when genes get far apart, now there's more room in between the 2 genes for crossing over to occur. So what happens is sometimes you get a crossover, and then sometimes in the same cell you get another crossover. And we call that a double crossover. And when you get a double crossover, there's actually several different ways to get double crossovers, and we'll go over those later. But if you get this kind of double crossover, what happens is you get this one, which is this, which is a parental. You get this one down the bottom, and that's also a parental. And that's because whatever genes start out together, if they stay together after recombination, then they're called parentals. But now what happens with this guy? So this guy, we get the big A, which should be blue. So we got this. Then we cross over. So if we drew the chromosome, we'd start out with some blue. Then we'd cross over, and we'd get some red. And then we cross back over up here, and we get blue again. And there's the big A's on here, and then we come up and get this blue again. We got little b over there, or big B over there. On the other side, we get some red, some blue, and some red. But because we cross over twice, we actually take this little a, pick up some of this blue stuff, come back down, pick up some of this red stuff. And even though the chromosomes look different, so recombination has occurred, if we look at the genes, because that's really the only thing we can look at, the genes look as if nothing ever happened. And so these guys show up as parentals again. So even though we've got more crossing over than we did up in the above example, so we're starting to get more crossing over, sometimes that crossing over regenerates the parentals. And so they don't get counted as recombinants. And because recombination distance equals percent recombinants, the way we figure out percent recombinants is the number of recombinants over the total recombinants plus parentals. If now the way we do this, we're generating more parentals, it's going to decrease the apparent number of recombinants. So this is an underestimate of the true genetic distance, because the true genetic distance is that they're really far away. But when we start getting more crossing over, now we start to regenerate those parentals, and it makes it look like recombination never occurred. Alright, so we'll see that again. But that's kind of how that goes. It's kind of a good question to get at that information. Alright, so this is getting into something called 3-point crosses. And so we'll talk about this in some detail because it's really related to those double crossovers that I was just showing you. So what happens if so let's redraw that scenario. And we get a double crossover. And since I know what I'm trying to make, we'll just do this. So the same thing that happened on the last little slide. And we got some genes on here. And we had a double crossover. And so it looks like nothing happened. So what could we do? So the problem is that in order to see what happened, we need to have a gene, because these letters represent genes that have a mutant phenotype that when we actually do a cross and we grow these things up into adult organisms, whether they're flies or whatever, we can tell which genes we got. We can tell what the phenotype is. This might have been black body and purple eyes that started out together, and then this might have been wild type and wild type, and then we kind of rearranged them. So even though we got crossing over, we can't tell that we rearranged these genes. So how can we tell? We can add another gene. And we'll make it yellow. So let's say we put a big, well, you can't barely see yellow, so let me get my chromosome back. Alright, let's make it, yeah, we can make it green. So we had big C, little c, little c. So now when we get crossing over, we'll still have a big C there and a little c there, but our C's in the middle will have switched places. myself here. So those genes are the same. So what does that look like when we do a cross? This is, we're saying we've got 3 genes. Let's keep our colors the same. Actually, so we don't. The thing is, we don't usually get these different colors. So the way this will look like in like an example would be something that's heterozygous for 3 genes, is test crossed, and if these genes are linked, we want to know which one is in the middle. And so I've drawn them like this, but we don't know which one's in the middle. I just sort of picked a random order. And then if we do this cross and we find out that we get some double crossovers, remember these are double crossovers, or DCOs. If we look at the double crossovers and we say the double crossovers look like this, even if we don't even have to put them in order yet. We'll put them like this, because this is the way it sort of looks in a problem. Is we know if we went from this configuration to whatever that is, to our double crossovers like this, the thing that changed, the thing that's different, is the thing that was originally started in the middle. So that tells us that what happened is we started out with that we can actually now put C in the middle, and we'll have to tell you how you figure out which ones are double crossovers. But what that is saying, and sometimes we'll draw it like this, and this is how they started, and then when you get a double crossover, they end up giving you this. This will be much more apparent when we do what's called a 3-point cross. So this question is actually much easier to answer if we have actual 3-point cross data. But this is what they're getting at, is that if you can identify which ones are the double crossovers, and you can compare those double crossovers to where you started. So here's where we started, and then these are the kinds of gametes that we got out from our double crossovers. That tells us which one's in the middle. Alright, so we're going to leave this one here, and then we'll go and do some of the problems, and some of these concepts will come back up. Actually, let me make sure there's no more. Yeah, so then we get into problems. So we'll stop this video here, and then we'll